Leicester Warwick Medical School. Cell injury. Dr. Gerald Saldana. Department of Pathology. Introduction. This presentation will be a guide to cell injury and cell death. Outline causes and pathogenesis of cell injury/death. Describe the morphological changes of cell injury/death. Describe the process of apoptosis. Introduction. General considerations. Adapt or die. Reaction patterns in a given cell/tissue is often limited. Degree of injury is a function of type, duration and severity of insult. Types of insult: hypoxia, ischemia, local, for example embolus, systemic, for example cardiac failure, hypoxemia, oxygen problems, for example altitude, hemoglobin problems, for example anemia, oxidative phosphorylation, for example cyanide poisoning. Types of insult: chemical. Many of the common poisons, arsenic, cyanide, mercury, interfere with cellular metabolism. If ATP levels drop below critical levels, affected cells will die. The list of pharmaceuticals that may have toxic effects on cells is enormous. Some act directly, but most have their effect through breakdown metabolites. Metabolism of alcohol, a type of drug, to acetaldehyde is one example. Types of insult: infections, fungi, rickettsia, bacteria, and viruses. For example, viruses can take over protein translation machinery and subvert it entirely to the production of new virions. Types of insult: physical. Direct physical effects. Exposure of tissue to extreme heat or cold results in direct injury that is often irreversible, resulting in a pattern of coagulative necrosis. See later. Sudden changes in pressure can cause cellular disruption, for example a hammer blow to the thumb. Electrical currents can cause direct breakdown of cellular membranes that may be irreversible. Types of insult: immune. Inflammatory mediators such as interferons and interleukins can alter both gene expression and cellular metabolism. The effects are designed to help cells combat an infectious process, but the resulting stress to the cells can be highly injurious and sometimes deadly. Activation of complement can result in direct attack on a cell's surface membrane. Cytotoxic T cells and NK cells can mediate a direct attack on a target cells and initiate the self-destruct cascade within a target cell. Types of insult: nutrition. Dietary insufficiency of protein, vitamins and/or minerals can lead to injury at the cellular level due to interference in normal metabolic pathways. Dietary excess can likewise lead to cellular and tissue alterations that are detrimental. For example, fat is the biggest offender, or excess ingestion of health supplements. Causes of cell injury. Summary: hypoxia, chemical, physical, infection, immune, nutritional deficiency, or excess. Principal structural targets for cell damage: cell membranes, plasma membrane, organelle membranes, DNA, proteins, structural, enzymes, mitochondria, oxidative phosphorylation. Pathogenesis of cell injury: hypoxia, reversible, loss of ATP, failure of Na/K pump, anaerobic metabolism, increased lactic acid and phosphate, reduced protein synthesis. Pathogenesis of cell injury: hypoxia, irreversible, massive intracytoplasmic calcium accumulation, enzyme activation. Pathogenesis of cell injury: general, reduced ATP synthesis/mitochondrial damage. loss of calcium homeostasis, disrupted membrane permeability. Free radicals. Free radicals highly reactive, unstable chemicals associated with cell injury. Chemicals/drugs, reperfusion injury, inflammation, irradiation, oxygen toxicity, carcinogenesis. Free radicals. Free radical generation occurs by absorption of irradiation. For example O and H. Endogenous normal metabolic reactions. For example O2 and H2O2. Transition metals. For example Fe+. Nitrous oxide. An important paracrine type mediator that helps regulate vascular pressure. Toxins. For example carbon tetrachloride. Free radicals. Free radicals are removed by spontaneous decay. Antioxidants. For example vitamin E, vitamin A, ascorbic acid, glutathione. Storage proteins. For example transferrin, ferritin, ceruloplasmin. Enzymes. Catalase, SOD, glutathione peroxidase. Free radicals injure cells by membrane lipid peroxidation. Autocatalytic chain reaction. Interaction with proteins. Protein fragmentation and protein-protein cross-linkage. DNA damage. Single-strand breaks, genomic and mitochondrial. General protective mechanisms. Heat shock response genes comprise a large group of genes. Expression is upregulated in the face of cell stressors and serve to protect proteins from stress-related damage. Clean up damaged proteins from the cell. Many tissues and organs can survive significant injury if they are pre-stressed. Ways to exploit this phenomenon to improve organ transplantation and tissue repairs are being tested in clinical trials. Cell injury, morphology. Reversible. Irreversible. Cell injury, morphology. Light microscopy. Cytoplasmic changes. Nuclear changes. Cell injury, morphology. Abnormal accumulations. Lipid. Protein. Necrosis definition patterns. Death of groups of contiguous cells in tissue or organ. Patterns. Coagulative. Liquefactive. Caseous. Fat necrosis. Gangrene. Infarct. Red/hemorrhagic. White. Coagulative necrosis. Cells have died but the basic shape and architecture of the tissue endures. Most common manifestation of ischemic necrosis in tissues. Affected tissue maintains solid consistency. In most cases the necrotic cells are ultimately removed by inflammatory cells. The dead cells may be replaced by regeneration from neighboring cells, or by scar, fibrosis, coagulative necrosis. Normal necrosis, coagulative necrosis. Liquefactive necrosis. Complete dissolution of necrotic tissue. Most commonly due to massive infiltration by neutrophils, abscess formation. Release of reactive oxygen species and proteases. Liquefaction is also characteristic of ischemic necrosis in the brain, liquefactive necrosis. Caseous necrosis. Accumulation of amorphous, no structure, debris within an area of necrosis. Tissue architecture is abolished and viable cells are no longer recognizable. Characteristically associated with the granulomatous inflammation of tuberculosis. Also seen in some fungal infections, caseous necrosis. Caseous necrosis. Fat necrosis. results from the action of lipases released into adipose tissue. Pancreatitis, trauma. Free fatty acids accumulate and precipitate as calcium soaps, saponification. These precipitates are grossly visible as pale yellow/white nodules. Microscopically, the digested fat loses its cellular outlines. There is often local inflammation. Fat necrosis. Gangrene, gangrenous necrosis. Not a separate kind of necrosis at all, but a term for necrosis that is advanced and visible grossly. Ipteris mostly coagulation necrosis, i.e., the typical blackening, desiccating foot which dried up before the bacteria could overgrow, we call it dry gangrene. Ipteris mostly liquefactive necrosis, i.e., the typical foul-smelling, oozing foot infected with several different kinds of bacteria, or if it's in a wet body cavity, we call it wet gangrene. Gangrenous necrosis. Infarction, an area of ischemic necrosis in a tissue or organ. White. Arterial occlusion in most solid tissues. Red/hemorrhagic. Venous occlusion. Loose tissues. Dual blood supply. Previously congested, white infarct. Red infarct. Apoptosis, basics. Is a distinct reaction pattern which represents programmed single-cell suicide. Cells actually expend energy in order to die. Derived from Greek falling off, as for autumn leaves. Apoptosis is the physiological way for a cell to die, seen in a variety of normal situations. Apoptosis, morphology. Necrosis. Pathological response to cellular injury. Chromatin clumps, mitochondria swell and rupture, membrane lysis, cell contents spill, inflammatory response triggered. Apoptosis. DNA cleaved at specific sites, 200 BP fragments. Cytoplasm shrinks without membrane rupture. Blebbing of plasma and nuclear membranes. Cell contents in membrane-bounded bodies, no inflammation. Apoptosis, normal. A stain for apoptotic cells in the developing paw of a fetal mouse. Apoptosis pathological. Graft versus host disease in colonic mucosa. Apoptosis, triggers withdrawal of growth stimuli, death signals. For example, growth factors. Death signals. For example, TNF and FAS. DNA damage. P53 plays an important role. All cells are equipped with the tools of their own destruction. The final common pathway in most instances of cell death involves the activation of cascades of cystine-type proteases called caspases. These cascades, which can be turned on through a variety of mechanisms, lead to the fatal destruction of proteins, lipids and nucleic acids, resulting in the irreversible signs of cell injury noted in the previous page. Apoptosis, mechanisms. Extrinsic factors. For example, by members of the TNF family. Intrinsic mechanisms. For example, hormone withdrawal. The two major pathways for caspase activation in mammalian cells are outlined. The extrinsic pathway, left, can be induced by members of the TNF family of cytokine receptors that are present at the surface of many cells. Upon activation, these receptors recruit adapter proteins to their cytosolic death domains, DD, including FAD, which then bind and cleave procaspases, particularly procaspase 8. The intrinsic pathway, right, can be induced by various stimuli, discussed below, resulting in the release of cytochrome C from mitochondria. In the cytosol, cytochrome C binds and activates APOF1, allowing it to bind and activate procaspase 9. Active caspase 9, intrinsic pathway, and caspase 8, extrinsic pathway, have each been shown to directly cleave and activate the effector protease, caspase 3. Other caspases can also become involved in these pathways. The two pathways are not mutually exclusive, both may become activated in response to an insult. Thus, this schematic represents an oversimplification of the events that occur in vivo. Summary: this talk has covered causes of cell injury, cellular targets, pathogenesis, morphology of cell injury, patterns of necrosis, apoptosis,